Good morning. My name is Tejaswini Kumawat and I will be teaching today topic photosynthesis in a higher plant for NEET botany. So what is photosynthesis and what we understand from the term photosynthesis? Photo means light and synthesis is production. So synthesis in the presence of a light. So what is synthesis? The synthesis is plant is synthesizing their food, that is glucose, which is further stored in the form of a starch. And how they are doing their process of photosynthesis? So simply we can see here a plant which is utilizing the light energy from the sun, CO2 from the air and the water molecules from the soil, and then they are doing the process of the photosynthesis. In further video we will learn the detailed process, and they are releasing the byproduct in the form of oxygen. So thus they are doing the photosynthesis for releasing the oxygen? No, they are doing photosynthesis for synthesizing their own food, that is a glucose, which is then stored in the form of the starch and releasing oxygen as a byproduct. Further, what are the basic requirement of the photosynthesis process? The basic requirement is light, which is gained from the sun, CO2, which is derived from the air, water which is taken from the soil, and chlorophyll, the photosynthetic machinery which is present in the mesophyll cells. Okay, so these are the four major basic requirement of the plants for the process of the photosynthesis. When time before, when we don't know the whole machine, the mechanism of the photosynthesis, the scientists were doing the basic experiments to know that they are actually required, these basic substance are actually required in the process or not. So the first experiment was variegated leaf experiment. In that experiment, the leaf was half covered with a black paper. So if the exposed leaves were able, in the presence of the light, food, CO2 and the water, were able to perform the process of the photosynthesis, but the part of the leaf which was covered with a black paper was not able to do the process of the photosynthesis. So there was no starch synthesis in that particular part of the leaf. So from this experiment we can able to understand that the light is a necessary for the process of the photosynthesis. Second experiment was KOH soaked cotton experiment, in which a leaf was taken, a plant was taken in a test tube, and a test tube having a KOH. So KOH will going to absorb the CO2, which is slight acidic, and KOH is slight alkaline. A neutralization reaction, KO2 will absorb all the CO2 from the surrounding air and will form K2CO3. So now no CO2 will be available in the surrounding area, and there will be no process of the photosynthesis in the leaf. As no CO2 is available, the plant will not able to synthesize their own food. Third experiment was a bell jar experiment. Bell jar is a glass jar. So the scientist Priestley performed the bell jar experiment, and how he do that experiment? He first kept a bell jar, a mouse under the bell jar, and after 2-3 minutes the mouse feels suffocated. Then he placed the candle below the bell jar. The candle was extinguished, and in the third experiment he placed mouse, candle and a mint plant in the bell jar, and they all were stable. How they were stable? Because the plant releasing the oxygen which was used by the mouse and the candle for the process of their respiration, and vice versa, the plant was using the CO2 for released by the mouse and the candle for the process. In the next experiment, that is Inselman experiment, he placed the aquatic plant under the sunlight. In same aquatic plant, in the dark, and what he observed that in the presence of the sunlight the plant was releasing the bubbles, and in the dark the bubbles were not released, and later he identified that the bubbles were of oxygen. In the next experiment, said that the glucose is produced in the starch, glucose is produced in the plant in the process of the photosynthesis, which is being stored in the form of the starch, and the chlorophyll is present in special bodies. And what are that special bodies? That special bodies is a chloroplast, which is present in the mesophyll cells where the process of photosynthesis is occurring. And then the next most important experiment is Engelmann experiment. In Engelmann, take the Cladophora algae and he placed a prism and illuminated a Cladophora with a white light. The prism divide the light into a 7 color, that is Vibgyor, and he observed that the accumulation of an aerobic bacteria in the blue region and in the red region. Why the bacteria were accumulating in that region? Because the maximum the process of the photosynthesis were occurring in the blue and red region. So in that area maximum oxygen were releasing and the maximum aerobic bacteria were accumulating there to utilize that oxygen. And from this experiment he described the first action spectrum of the photosynthesis. So what is action spectrum? The action spectrum of the photosynthesis is in what region, in what wavelength, where the maximum photosynthesis is occurring. So from this chart, this graph, we can understand that the maximum photosynthesis is occurring in the region of 4 to 500 nanometer, and then in 600 to 700 nanometer. We will going to see this graph further. Next experiment is the Neal experiment. When Neal experiment, he had studied the green and purple sulfur bacteria, where he said that the hydrogen from a molecule is required to reduce the CO2. A molecule having an hydrogen will, if able to reduce the CO2, then only the organic molecule will be produced, and depending upon the what the molecule is taken from the hydrogen, depending upon that the product will be formed. If this molecule is H2A, if it is going to H2O, then oxygen is released. But where in green sulfur bacteria and purple sulfur bacteria, if this H2A is H2S, hydrogen sulfide, then instead of being the oxygen, the sulfur will going to release, and that process is known as an oxygenic photosynthesis, because there the product will be sulfur instead of being oxygen, because the compound is taken as hydrogen sulfide, not H2O. Now what is the structure of chloroplast? What is the basic structure where the photosynthesis is machinery is present and the whole process of the photosynthesis is occurring? We will going to learn the structure of the chloroplast. The chloroplast consists of an outer inner membrane. Then this stack-like structure, this is known as a single structure, is known as a thylakoid. And when a single thylakoid, there is a stack of the thylakoid, then this is known as a grana. Grana, a whole group is known as a grana, and a single is known as an thylakoid. In the thylakoid, lumen is present, and a thylakoid is surrounded by a liquid, that is a stroma. The one grana can be connected with another grana by a stromal lamellae. What do we understand by the term lamellae? Lamellae means membranous structure. So as it is surrounded by the stroma, this connecting structure is known as a stromal lamellae. So chloroplast, as we know, has its own nucleic acid. It has 70S ribosome structure for synthesizing its own protein, as it is a synthetic semi-synthetic organelle, so it has in starch granules. As it is doing the photosynthesis, the glucose is being converted into the starch. It is being stored in the form of the starch granules. So the chloroplast has a thylakoid which is being stacked upon each other to form the grana. Then the grana or the thylakoids may be connected with each other by a stromal lamellae. So this part is a stroma. The liquid part is known as the stroma. The space inside the thylakoid is known as the lumen, where in the membrane of a thylakoid the whole process of the photosynthesis is occurring. And how it is occurring, we will see it up further. Now we will read about the absorption spectrum. What is an absorption spectrum? So as we all know, the electromagnetic spectrum, which has an gamma rays, X-ray, UV rays, visible, microwave, radio waves. In that spectrum, only the PAR region, that is photosynthetic active region, which is a visible region, which is 400 to 700 nanometer range. In that particular region only the process of the photosynthesis is occurring. So the plants have different pigment molecule, that is chlorophyll A, chlorophyll B, carotenoids, xanthophylls. So the major pigment which is responsible for the process of the photosynthesis is chlorophyll A, which is giving the maximum absorbance between 400 to 500 and then 600 to 700. Then other pigments also we have chlorophyll B, carotenoid, which have their own wavelength of the maximum absorbance. So the pigments other than the chlorophyll A are known as accessory pigments because they help the chlorophyll A in the process of the photosynthesis. So in this chart we can see that where the maximum absorbance has been shown by the chlorophyll A in the region of 400 to 500 nanometer and between the 600 to 700 nanometer. In that particular area the action is performed. What is the action? The photosynthesis, the process of the photosynthesis is being occurring in the same particular range of the wavelength. So this is the absorption spectrum. Absorption spectrum, that is the maximum range of the wavelength where the chlorophyll molecule, the chlorophyll A molecule absorbs the light. In that particular area the process of the photosynthesis occurring. So the absorption is occurring, that is known as absorption spectrum. In which range absorption is occurring maximum? In the range of 400 to 500 and then 600 to 700. And in that particular wavelength the process of the photosynthesis were maximum. So this is a action spectrum and this is a absorption spectrum. Why the different molecules, the pigments are present in the plant for the photosynthesis? Because they have their own particular range of the wavelength where they are performing, where they are gaining the light, they are absorbing the maximum energy and then they transfer that particular energy to the chlorophyll A. And even when the light intensity is so very high, then the accessory pigments molecules like the carotenoids help help and inhibit the process of the photooxidation, so that our main pigment, that is chlorophyll A, is not oxidized, is not oxidized, and the process of the photosynthesis can be continued. So the other pigments are absorbing the light energy in from the range of the 400 to 700, and the main pigment is a chlorophyll A molecule. Thank you. Till now we have learned that the chlorophyll A is the important pigment for the photosynthesis, and other pigments like the chlorophyll B, xanthophylls and the carotenoids are the accessory pigments which absorbs the light and transfer the energy to the chlorophyll A. Now we will going to learn what are the photosystems. Photosystems is there are two photosystems, photosystem 1 and photosystem 2. The photosystem 1 is composed of a light LHC, that is light harvesting complex, and the reaction center. The reaction center is the center from which the process of the photosynthesis is the start, from the where the whole process of the electron transfer is started. And this reaction center absorbs the maximum energy, maximum wavelength of the light, that is of 700 nanometer far red light ray. And what is light harvesting system? The light harvesting complex is made of the chlorophyll A and accessory pigment molecules, which will going to utilize the sunlight from the, and they will transfer the energy to the chlorophyll A molecule. Each of the pigment accessory molecule will going to utilize the light energy, and they will transfer that particular light energy to the special chlorophyll molecule A, that is known as a reaction center. And what is the difference between that particular chlorophyll A and the other pigment is that they will going to transfer the energy, but the reaction center chlorophyll A will going to transfer its electron to the primary electron acceptor. From there, the whole process of the electron transfer will take place. So the pigment molecule will absorb the light energy. They will transfer the light energy to the chlorophyll A molecule, special chlorophyll A molecule, that is known as a reaction center, which will going to absorb the light of the 700 nanometer, and when it is in the excited state, it will going to transfer its electron to the first primary acceptor of electron. From there, the whole process of the photosynthesis, the whole process of the electron transfer will going to start. Then we have photosystem second. Same as a photosystem 1, we have the light harvesting system complex and the reaction center, which will going to absorb the light of the 680 nanometer, that is in the range of a red light. So reaction center of a photosystem second is also a special chlorophyll A molecule, which will going to transfer the electron to the acceptor. But it has, that is the light harvesting complex, will have the pigment molecule, the accessory molecules, which will transfer the energy to the chlorophyll A molecule. So, and how the process of the whole process is occurring, now we will going to see. So our process of the photosynthesis is divided into two reactions: light dependent reaction, and second is a dark reaction or Kelvin reaction. So what are the light dependent reaction and what is Kelvin reaction? In short, we will going to see here in this diagram. So we have in the chloroplast diagram, we have learned that it has n thylakoids, which in the form of a stack upon each other will form the grana. So each thylakoid or grana, by in the light reaction, are synthesizing some of the energy molecule, that is ATP, NADPH, and releasing oxygen as a byproduct. That ATP and NADPH is being utilized in the dark reaction, that is a Calvin cycle, for where each molecule from the carbon dioxide from the air is being fixed and will converted into a triose phosphate for the production of a glucose molecule and then further starch. So ATP, NADPH from the light reactions are being utilized in the Calvin cycle dark reaction for the fixation of the carbon dioxide molecule from the air. This Calvin cycle is also known as dark reaction, not that because the reaction is performing in the dark, but because it is not directly utilizing the light. It is not utilizing the light, but it has utilized the products of the light reaction, that is ATP and NADPH But the light reaction is utilizing the light, and how it is utilizing the light we can see further. So we will learn the process of the non-cyclic photophosphorylation and cyclic photophosphorylation. So non-cyclic photophosphorylation, as we learned that we have two photosystem, photosystem 1 and photosystem 2. Photosystem 2, the whole process of the non-cyclic photophosphorylation starts with the photosystem 2nd and then with the photosystem 1. So photosystem 2nd is consists of a reaction center 680 which will utilize the light of a 680 nanometer. So when the light falls of a 680 nanometer, 680 nanometer, the reaction center get excited. The accessory pigments will transfer the energy to the photosystem 2nd. Photosystem 2nd get excited and will transfer its electron to the first primary electron acceptor. And what is that acceptor? The first acceptor is a pheophytin. So when a photosystem 2nd will transfer the electron to the pheophytin, that is a primary electron acceptor, itself get oxidized, but it will going to reduce the pheophytin. So what type of the reaction that is? It is a redox reaction. Then the pheophytin will then transfer its electron to the plastoquinone, then plastoquinone, pheophytin get reduced, plastoquinone get pheophytin get oxidized, plastoquinone get reduced, then the plastoquinone will further transfer the electron to the cytochrome B6F complex, then the cytochrome B6F complex transfers the electron to the plastocyanin. Same way the cytochrome B6F get oxidized, plastocyanin get reduced, and the plastocyanin then finally transferred its electron to the photosystem 1, that is P700. Then P700 will gain electron. It's a special type of the chlorophyll A molecule, as we learn in the photosystem. It will then again transfer, when it is in the excited form, it will again transfer its electron to the primary acceptor, that is A0, which an special type of the chlorophyll. Then from there the electron will be transferred to the iron sulfur cluster. From then the ferredoxin molecule, the electron will be transferred to the ferredoxin. From the ferredoxin, then transferred the electron to an enzyme, that is ferredoxin NADPH reductase, and this enzyme will transfer its electron to the NADP. And when the NADP will get the electrons plus hydrogen, they will convert into the NADPH plus H. So what is NADP? Nicotine amide adenine diphosphate. It will get the electron from the FNR, ferredoxin NADPH reductase enzyme. So this is the whole process of the non-cyclic photophosphorylation. Why it is non-cyclic photophosphorylation? Because the electron is being given by the photosystem 2nd, but it is not again transferred to the photosystem 2nd. Instead, they are gained by a molecule NADP. So how then? How if it has lose its electron, how it will gain its electron. So find the molecule from which it is gain its electron is in water molecule. Water molecule in the lumen will be splitted into H plus, electron and oxygen. So it will transfer its electron to the photosystem 680, P2 photosystem. So this complex is known as oxygen evolving complex. It will transfer its electron to ultimately to the 680. So the ultimate electron acceptor is NADP, and the electron molecule which gives the electron is a water molecule. This is the whole process of a non-cyclic photophosphorylation. Now what is a cyclic? So what are the products of the non-cyclic photophosphorylation? The four products are NADPH, ATP, and we will going to learn how the ATP is produced and the oxygen. So we have seen the oxygen and we have seen the NADPH. Now the process, we will learn the process of the cyclic photophosphorylation. So in the cyclic photophosphorylation, the process is start when the lights fall directly on the P700. So actually the light is falling on both the photosystem P700 and P680 also. So when the light fall on the P700, it will get excited and will transfer its electron to the primary acceptor. Then the primary acceptor will transfer the electron to the iron sulfur clusters, then to ferredoxin, and then from the ferredoxin the electrons will be transferred to the side. So the plastoquinone will transfer the electron to the cytochrome B6F, then the cytochrome will transfer the electron to the plastocyanin, and then plastocyanin will give again the electron to the photosystem first. So this is the whole process is a cyclic. The ultimately electron was given from the P700 and ultimately the electrons were again accepted from the P700, and in the process the ATP is produced. Only ATP is produced, as the FNR enzyme is not present in this cycle, so NADPH will not be produced. Only ATP will be produced. And why oxygen is not oxygen is not produced here? Because the oxygen evolving complex is present, is associated with the photosystem second, and in this process photosystem second is not present. Only photosystem first is present. So the whole reaction is a redox reaction. When the P700 give its electron to the primary acceptor, it will itself oxidize and will reduce the primary acceptor. When primary acceptor will give the electron to the iron sulfur clusters, then the iron sulfur cluster will get reduced and primary acceptor will oxidize. So it is a series of an redox reaction where a molecule get oxidized, reduced, oxidized. So here the most important thing is the molecule is that a plastoquinone. The plastoquinone, when it's transfer the electron along with an electron, this molecule will transfer the hydrogen ions from the stroma into the lumen. So into the lumen, the concentration of the hydrogen ion will increase. There are the three steps where in the whole process of the non-cyclic photophosphorylation will going to increase the concentration of the protons, the hydrogen ions in the lumen as compared to the stroma. That we will going to learn in this diagram, and we will understand more detail in this process. So the plastoquinone plays an important role. It will transfer the hydrogen from the stroma into the lumen, and as we have seen, when the water is splitting, it will convert into an hydrogen ions, electrons, and O2 being a gas is released into an environment and hydrogen ions is released. So the protons automatically will, if it really is splitting, then the concentration of the hydrogen ion will going to increase in the lumen. So this particular step going to increase the concentration of the protons. When the electrons are transferred from the plastoquinone, the proton molecules from the stroma will going to pass from the stroma into the lumen. This step going to increase the concentration of the proton, and when NADP plus is being converted into NADPH plus, then the hydrogen from the stroma are utilized to being converted into a reduced form. So these three steps are the major steps which will going to increase the concentration of the proton ions in the lumen as compared to the stroma. Now we will going to learn the chemoosmotic hypothesis. So how actually the production of ATP is occurring, why we are focusing on the transfer of electrons from the one molecule from the photosystem 1 to the photosystem second, and then the transfer of the electrons to the FNR for the production of the NADP. The most important process why we are we are reading, this is the synthesis of the ATP and how these processes are involved in the synthesis of the ATP. So in, as we have learned in the non-cyclic photophosphorylation, there are the three steps way which are going to increase the concentration of the protons in the lumen as compared to the stroma. The first process is when the photosystem, second is transferring its electron to the primary acceptor, that is the pheophytin, which will transfer the electron to the plastoquinone. Plastoquinone, when it transfer the electron to the cytochrome B6F, so along with the electron, it will transfer the hydrogen ions from the stroma into the lumen. This is the first step which will going to increase the concentration of the hydrogen ion. Then the water splitting, the splitting of the water and the non-cyclic photophosphorylation, when the photosystem second is get oxidized, where does it will gain the electron? It will gain its electron again back from the water molecule, which will going to release the proton ions. So these proton ions will going to increase the concentration of the protons in the lumen. And the third is, these are the second step, and the third is that when NADP plus is being reduced to the NADPH, it will utilize the hydrogen ions, the protons from the stroma. So this will decrease the concentration of the hydrogen in the stroma. So from this step, the hydrogen ions concentration increases in the stroma, which will generate the electrochemical potential or electrochemical gradient. As a high concentration of the protons are present in the lumen, this will lead to the generation of the positive charge, slightly a positive charge along the membrane, and a negative charge, positive charge inside the membrane, in the inner side of the membrane, and a negative charge outside the membrane due to the difference in the hydrogen ion concentration. So we can see from here, the concentration is low in the stroma and the concentration is high in the lumen. So we will now learn about this ATP synthetase. ATP synthetase molecule. ATP synthetase molecule is consist of CF0 particle which is embedded in the membrane and CF1 particle. So why it is known as CFO? Because it is inhibited by an antibiotic oligomycin, and CF0 will allow the transfer of the hydrogen ions and a proton motive force. Proton, from the movement of the proton, the energy is released. The protons will move from the CF0, CF1, the ADP and PI. When the electron will transfer, it will rotational movement will occur in the CF1 and it will be converted into ATP. So here the whole process of the ATP generation take place. So we will have learned till now that in the non-cyclic photophosphorylation, the three products are NADPH, ATP and O2. How oxygen is synthesized? Oxygen is synthesized from the oxygen evolving complex, from the splitting of the water molecule, NADPH from the FNR molecule when the whole process of the non-cyclic phosphorylation occurring, and ATP from the when the proton motive force is proton constant gradient is generated and electrochemical gradient is generated, and then the proton will move from the high concentration towards a low concentration from the ATP synthetase, and the rotation of the ATP when the movement of the hydrogen molecule is occurring. Rotational movement is occurring in the ATP synthetase, and which will lead to the generation of the ADP from ATP to ATP. So these three products are then utilized in the dark reaction, that is in the Calvin cycle. And now we will going to learn how these products are utilized in a dark reaction for the glucose molecule, for the production of the triose phosphate, for the glucose, for the starch molecule. In further we are going to learn, and this chemoosmotic hypothesis was identified by Peter Michel. Thank you.